Okay guys, welcome back. So in the last video, we took kind of a journey through the history of DNA and how we really figured out that this is going to be the important molecule that we need to look at for passing traits on from offspring to parent or that horizontal transfer. This is the information. So we also, after learning a little bit more about the structure, reviewing what that meant, talked about some of the experiments Vesselson and Salk sort of performed so that we understood that DNA is going to replicate in a semi-conservative way. So in this video, we're going to take what we knew from the last video and we're going to really start to get into the mechanics of replication. Okay, so let's start out by just talking briefly about replication as it happens in a prokaryotic cell. Okay, so some things to note, of course, in prokaryotes, we have a circular piece of DNA. In eukaryotes, this is not going to be the case. That'll have some ramifications later, but we'll get to those. Okay, so we can learn a lot about replication and how it's going to get started from just thinking a little bit about prokaryotic cells. So in their circular piece of DNA, note it's still double-stranded, they're going to have a specific DNA sequence, okay, a specific sequence that's going to be called the origin of replication. So this will be in bacteria, the single sole place where replication starts. So notice what's going to happen is we're going to start synthesizing a new strand, creating what's called a replication bubble with a fork on either side. So the whole thing is the bubble. The fork is just the kind of Y, where this kind of Y is open here. Okay, and it will proceed in both directions until we've completely replicated both strands, and now we're done. Okay, this is a really cool microscopy image. This is electron microscopy. We're looking at a replicating bacterial chromosome or piece of DNA. This whole thing here is the replication bubble. So this is not quite, but maybe almost halfway along. Okay, so the DNA is double-stranded here, it's double-stranded here, it's double-stranded everywhere. Okay, except for exactly where it's actively going to be open. And each one of these arrows is the fork. Okay, so that's literally what it looks like. So again, in bacteria, this happens at one spot. It's going to start at one spot and then sort of progress. In eukaryotic cells, we're going to have origins of replication kind of interspersed throughout a longer linear chromosome. Okay, so we can create multiple bubbles at once. Each of these bubbles has its own set of forks, one on each side, and these are going to expand as we create new DNA. That fork will proceed in this case, the right-hand direction, in this case, the left-hand, until they meet up, and then they become a solid DNA molecule, this new daughter strand. Okay, so you can see a similar kind of thing here. This is a eukaryotic chromosome. You can see multiple bubbles. Here's one bubble, here's another bubble. Okay, so here's a piece where it hasn't gotten to it yet. Okay, fork on this side, fork on this side, fork here, fork there. Okay, so you can see multiple places that this is happening. All right, and this is going to, again, have some ramifications, the fact that this is linear, but we're not going to see those just yet. Okay, so before we do get into what exactly is happening, we're going to be really deep in these replication bubbles here in a second. Before we get into that, it's important to sort of put this into a more global context. So it's really, really important that we understand that replication of DNA happens for one reason only. That cell is about to divide. So in our eukaryotic cell, we are preparing for mitosis. This is S phase. That is the only time we should be replicating our DNA. Okay, so as humans, we have 23 chromosomes. You're going to have 2 copies of each of them. You do not need more copies of that to live a regular existence. You only replicate your DNA when you're about to undergo mitosis. That is 100% the only time. Okay, so I'm making that really clear. Tell that to yourself. whenever you need to, because when we start talking more about the central dogma in chapter 17, transcription translation, that's going to be a whole different story. We're going to be deep into some DNA there, but it's going to be a different situation. So replication is only for cell division. That is it. Okay? So if you're making extra DNA, you do not need to be doing so. Okay, just for living a single cell life. All right. So as we get started, the way that this will kind of go is I'll introduce you to some of the replication machinery, give you guys a heads up of the important things we'll be looking for. We'll go through it in kind of a static way, and then I'll have a second video for you that you can watch that's a little more animated, so you can hopefully see how this progresses a little easier, okay? If you don't like the video I've picked out, just remember it's okay to Google and find something that really helps you visualize what's going on here, okay? So the important players, these are all enzymes that we'll be talking about, okay, except for the single-stranded binding proteins. So just keep an eye out, okay? They're loosely-ish in the order that we'll see them, although a lot of these things are happening all at once. All right, so helicase, we'll be looking at our single-stranded binding proteins. All right, make yourself a list, and you can make notes as you go that we're going to want to know what each of these does, okay? So let's get started. We're going to do eukaryotic replication. Step 1 starts with helicase and the single-stranded binding proteins. Okay, so helicases are a type of enzyme that help pull apart these hydrogen bonds. So they're not breaking anything, they're pulling the two strands apart. Hydrogen bonds are very weak on their own, but when you have a lot of them in a row here, this is not just going to come apart under normal physiological conditions. Okay, so we're going to use helicase to really help move that along. Okay, so moving our helicase to open up these strands, and that's going to be important because each strand we'll be using as a template. Okay, so we got to have these open. The single-stranded binding proteins kind of act just to hold this replication fork open until we get the replication done, okay? Because if we open this up, they're just going to want to snap back together if they don't have any reason that they should stay open. So until we get it replicated, we're going to need these DNA binding proteins to stay in place to hold this bubble open, okay? So replication actually happens very fast. It's going to happen in, a few hours in our cells, even though we have a ton of DNA. Okay, so this is going to just briefly kind of help us get everything started. Okay, so we've already seen two of our several players. Okay, and we'll talk more about helicase as we get into some of the details. All right, so let's take a closer look at what's going on. Okay, so hopefully you've noticed some things here. Okay, just take in the anatomy of this. There's a lot going on. Truthfully, it's happening simultaneously for the most part. It's very rapid. But we got to talk about it one piece at a time. So try to start putting this together in your mind. This is going to be simultaneous for the most part, but we're just talking about them separately. Okay, so note, now we've got some idea about the orientation of our DNA. The top strand, 5' on the left, 3' on the right. Our bottom strand, 3' on the left, 5' on the right. Okay, so anti-parallel. Just take a look, relax yourself. Two of these things we already know about. This is the left-hand side of the replication bubble. This is the fork that we're going to actively be working in. Okay, the helicase has already broken apart those hydrogen bonds, and the single-stranded binding proteins are holding everything open because notice there's no partner here. There's nothing to hydrogen bond with. It's going to want to snap back on. Okay, so We've already kind of talked about these, but now we've been able to position them a little bit more clearly with what's going on here. Okay, let's talk about our topoisomerase next. Okay, so we may have a description coming in. I know primase is what I've got down here, but I think this relates very strongly to the action of helicase. Okay, so helicase, it's going to open these double strands up. The way this is pictured, this image, the DNA is kind of linear, it's flat. Remember, this is a helix. They're wound around each other. If you start to pull them apart, it'll be like trying to pull apart two twisted threads. It'll just get more and more coiled. Okay, so this can happen to DNA as well, which can result in breakage and damage in an uncontrolled way. And it's referred to as supercoiling. Okay, so the tension of opening up these bonds and not allowing them to come back together, because again, single-stranded binding proteins and ultimately a new daughter strand, that is going to cause a lot of tension further down. Okay, to alleviate that physical tension, all right, we're going to have topoisomerase come in. It's going to cut the strands, allow them to untwist, and then seal it back together. Okay, so it's kind of allowing for that tension release. So really, if anything, what 2020 needs is a little bit more topoisomerase. Okay, so we cut the strands, allow them to untwist, stick them back together, and that alleviates any tension on the downstream strand. So it's kind of working ahead of helicase. So imagine these two guys moving together, going towards the left-hand side of the screen. On the other side of the fork, you'll have another topoisomerase, another helicase opening that up potentially. Okay, so we're working on that other side of the fork. Okay, but this is just the left-hand side. All right, so we've already talked about topoisomerase. Okay, I may have a note about that a little bit later, but I think it's important to kind of add that in right here because of the action, related action of helicase. Now we're going to get to the actual making a new daughter strand. Okay, so everything else up until this point has been set up. Once these two strands are apart, an enzyme called primase comes in and adds what we call a primer. Okay, so a couple of things about this primer. First of all, for whatever reason, our DNA polymerase that we're going to talk about soon, this cannot just come in and start dropping nucleotides down. Okay, I know it's mind-boggling. It seems like you should just be able to, like, oh, I can start matching this anywhere. Biochemically, for whatever reason, that is not what DNA polymerase can do. However, primase is totally fine doing that, so I don't know what's up with that, but that's just the way that it is. Primase can just start dropping nucleotides down when it sees an open place to do so. And this primer will allow us, when DNA polymerase finally does appear on the scene, to make this new daughter strand. This will give us something to latch on to. Okay, so that's the point, is we're priming, just like primase, we're priming the situation for DNA polymerase. Okay, the next thing we need to know about this primer is that it is not composed of deoxyribonucleic acid or DNA nucleotides. It is composed of RNA. So yeah, RNA, just like we talk about with mRNA in a similar way. Okay, it's not mRNA, but it does have ribonucleotides. Okay, that will have some ramifications later. Again, there's a lot of things later that will happen. All right, so I recommend looking at these, watching the video that I'm going to post next, plus the, anything that you want to look up on your own to see if there's, videos that might suit your learning style better. Try to start visualizing this as a fluid thing. Okay, it's not necessarily step by step by step. It's one fluid. We're opening this, we're unzipping it, just like pulling a zipper down, okay, from a jacket. It's not like I'm gonna do it like one click at a time. It's like unzip. Okay, and as it's being unzipped, topoisomerase is working ahead, making sure that this doesn't become too torqued out. Single-stranded binding proteins are there as long as there is no new daughter strand. This is single-stranded. Okay, and as this opens up, our RNA primer is added, and we are now ready for the situation. Okay, so we're almost there where we can add in the real deal, but we got to know some things about the mechanics. Okay, all polymerases, which primase is included, okay, in this particular situation, including the DNA polymerases we'll talk about today. Okay, anytime we're going to make a new DNA, RNA, anything, they are always synthesized starting at the 5' end to the 3' end. This is called 5' to 3' direction. What that means is you cannot add to the 5' end. You cannot add to it. You can't. Okay, you cannot do it. Only add to the 3' end. The result of that is that from this primer, notice it's anti-parallel with its parent strand 2. This primer can only go to the left. Okay, you can't do anything about the right now. Okay, we're going to figure out how that gets solved in a minute. For right now, wherever this gets dropped, you go 3'. That's it. Okay, it's anti-parallel with the template strand it's working on. All right, we go to the left. Okay, if we were on the bottom strand, our 5' end would be on the left-hand side. Our 3' end would be on the right to be anti-parallel with it. We'd go to the right. Okay, we're not working down here right now, but that's the setup. Okay, so the top things that you'll need to be acquainted with, if you just relax, make sure that everything you do is anti-parallel at all times. If you have 2 sets of nucleotides hydrogen bonded together, make them anti-parallel. If it's a primer, it's anti-parallel. If it's a new daughter strand, anti-parallel. If it's the old parent strand, anti-parallel. If you're able to do that, and just remember that you're only going to add to the 3' end, it will make your life so much better. Okay, so let's now add in that polymerase. All right, here's step 3. So in our eukaryotic cells, for the large part of DNA replication, this is going to be performed by DNA polymerase 3. Yes, it's important to know that it's 3 because we're going to have another type of DNA polymerase coming up. Okay, so look, we're okay right now. Helicase, opening up the strands. See it go? Single-stranded binding proteins, note, they're only around just in the single-stranded region. They're not up here where the daughter strand. This is not single-stranded anymore. It's got a daughter strand. It's a double helix, okay? Here is our primer. Primase just put this down for us. We don't need it here anymore. We'll go somewhere else and put a primer, okay? We're ready to go. Remember that we can only add on to the 3' end, which on this top strand is the left-hand side. We can only add going to the left. DNA polymerase works the same way. Here we go. Okay, and you may notice that this is called a sliding clamp. Okay, notice that this is in like 2 pieces. It's kind of like if this piece here was one of your hands. Okay, this is kind of the other of your hands. If you like wrapped your hands around like a pole, or alternatively a giant Route 44 Sonic beverage. Okay, like this is just like, they call it a sliding clamp because it's going to slide and it's kind of clamped onto the DNA. So nothing to worry about there. I probably, even though this is accurate, I probably won't mention it again right now for the rest of this chapter, but just so you know why it's here. Okay, so this is DNA polymerase. Its job, if we say it in the most simple terms, is to add new nucleotides. Okay, it is matching, you know, whatever. If this is a T, it's like I got to put an A here, and it's doing it and it's moving on. If this is a C up here on the parent strand, it's putting a G. Okay, so it's gonna match up the complementary nucleotide, and it's gonna zip along. This is fast, my friends. Like, you got to think about it fast. Okay, and it is going to be synthesizing new nucleotides. Okay. Aside from matching up the complementary base, which is what it's doing, notice it's also sealing that backbone, that sugar phosphate backbone. It is enzymatically what it is doing, aside again from matching the nucleotide to the complementary base pair, it is creating phosphodiester bonds. Okay, that is the bond that is going to be present in the backbone of DNA. So let's take a closer look at that. Okay, so here is our template strand. Okay, so this is the one that was already formed. Here is the growing strand. Note we can only add onto the 3' end. Okay, that is it. This is all we got. What we are doing is taking nucleotides, specifically their nucleotide triphosphate. Okay, so this would be like thymidine triphosphate. Okay, or if we're putting in adenine, this would be adenine triphosphate. Yeah, ATP, man, we use that for everything. Okay, guanine triphosphate, cytosine triphosphate. This is the raw nucleoside that we're getting ready to put in. Okay, so we also abbreviate these just so that you know. These little pieces, these nucleotides, these are like deoxydenosine triphosphate, deoxyguanine triphosphate. Okay, so that's what we're talking about here. Deoxy because it is DNA. All right. And collectively, when we got all 4 of them, we call them deoxynucleotide triphosphates. That's the whole group of all 4 of them, so a whole pool of everything. Okay, so we've got a lot of jargony things here, but like, check it out, man. We know that we have in this backbone sugar, phosphate, okay, and that phosphodiester bond that's happening. Okay, sugar, phosphate. So we're going to have, this is the sugar, we got to have another phosphate. That phosphate is this guy, okay? So we don't need these other 2. That's just going to help us power this reaction, sort of, because we, you know, negative phosphates don't really like to be together anyway, so we can boot 2 of them out. They just go wherever phosphates go, okay? They can go for oxidative phosphorylation. I don't really care where they go. They're away from me now, okay? These 2 are going to be gone, so this one here that's left will form a bond on this 3' end, okay? Then we've added the new nucleotide. All right, so that's this phosphate. Okay, so we had guanine, 3' end. Now we have guanine. We're gonna have phosphate, thymine, 3' end. Okay, so the phosphate came from here. Okay, we just swing that up, and now we've got a phosphodiester bond going on here and a new 3' end. So we can keep adding to that end only. So that is what's happening with DNA polymerase. It is doing it very rapidly, okay? It is matching the nucleotide, and it is catalyzing phosphodiester bonds. Okay, so we'll have another chance to think about that in a second. Let's do sort of a slow partial animation. Okay, hopefully this will set you up before you get to a more heavily animated video that I've posted for you. Okay, step 1-ish, okay, again, because some of these things happen kind of simultaneously, but we're still doing them. We're still talking about them one at a time. Helicase binds to our double strand and unzips our helices. So there goes our helicase, and they're apart now. Okay, so now what happens? All right, this is, let's say, 5' down here, 3' up here. Okay, remember that what's actually happening is we're splitting apart those hydrogen bonds. Okay, that's it. That's all that's happened. All right. So helicase did that. Our single-stranded binding proteins can be hanging out, keeping that open until our strand is doubled. Okay, so just holding it open. Primase comes in and lays down an RNA primer. Okay, so there you go. There's your RNA primer. And again, these will be the scaffold. Here, we have to synthesize towards the left because we can only add on to the 3' end. So if the 5' is over here, it's antiparallel. only adding in the left direction. Okay, so DNA polymerase comes in and it's going to match the nucleotide. So this is what we saw on the previous slide. I know it's backwards. Okay, I was just trying to match the orientation here, but you could just think this is, again, the new nucleotide that's going to be added, that phosphate swinging up. It's catalyzing that. DNA polymerase 3 is doing that right now. Okay, and it's only adding to the left-hand side as it goes. That's the direction it has to go. Okay, so that's all we've talked about. You should now really understand the function of most of what we've talked about, or at least that list I gave you. Not all of it quite yet, but a lot of it's covered. Okay, we got helicase, single-stranded binding proteins. We're going to have primase. We're going to have our DNA polymerase 3 and our topoisomerase. Okay, topoisomerase very much present in this example. It would be moving ahead of our DNA polymerase 3. Okay, so we only have a few enzymes left to really talk about. Okay, so you may notice we've only been working on the top strand for now, but we got to replicate that bottom one too. But that one's going to cause some logistical issues, so we got to get into that. All right, so things that we know: DNA is anti-parallel forever and always, and anything added to it, RNA, DNA, new anything, must also be anti-parallel. DNA polymerases can only create DNA in the 5' to 3' direction, meaning I can only add 2 to 3' in. That is it. That is what I have to do. So replication, because of this, is going to happen in opposite directions on the top and bottom strand. So everything that we've been working on before, if you notice, we were moving in to the replication fork. We were moving into the opening fork. We were going the same direction as helicase. Okay, but we left the bottom strand alone. Because of these directionality constraints, the bottom strand that we haven't touched so far will need to go to the right and out of the fork. Okay, so this will result in what's termed the leading strand, which is what we've talked about all up until now, on going into the fork, and the lagging strand, which will be going away from the fork. Okay, and is it really lagging? I don't know, man. They finish around the same, you know, they're replicating, they finish about the same time, but you'll see it just takes a little more effort. Okay, let's re-examine this replication bubble. Okay, replication bubble, replication fork, both are expanding at the same time. Up until now, let's stay on the left half of this because we've only been working on the left half. Okay, notice we're going to be working in this direction. Okay, so if you notice, there is a mislabeling here. I actually did not mislabel this. This was mislabeled. It is labeling both ends as 5' and 3'. Okay, so while anti-parallel is correct, okay, what's actually true is that this is the 3' end of the top strand. This is the 5' end. Okay, this is the 3' end of the bottom strand. This is the 5'. So just swap these guys. Okay, so sorry that that is there. I just noticed it as I'm recording this, but I didn't label it for once. A mistake that happened wasn't me. Okay, I made so many in my life and on most of this PowerPoint that I've been doing. So just think about switching these two and you should be better. Okay, so let's think about this. Okay, we've been operating on the left-hand side of the fork. All right, 5' over here. This is the same orientation we've been working in. This is your RNA primer put down by primase. Leading strand is going towards the opening fork. Think about it also the same direction as helicase. That means that as helicase opens this further down, we just keep adding nucleotides until we get to another replication bubble we can join with. Okay, we just keep going. But on the bottom strand, we gotta go the other way. Okay, so we gotta go 5' to 3'. Okay, on the left-hand side of this label correctly here, we have to add in the 3' side. We don't have a choice. or anything else. Okay, so think about it. We really just don't have a choice, man. Okay, we're going to the right-hand side. That means that we're going away from the fork. Notice, you already see there's a couple extra things going on here. Okay, extra primers. We're going to get to that. So the leading strand points into the fork. The direction of synthesis is into the fork or the same direction as helicase. The lagging strand, which is this bottom strand, points away from the fork or the opposite direction of helicase. So notice, though, we've been working on this left side. We're going to continue to do so. But just notice that when we cross over into the right half of this, things change. The guy that was the lagging strand over here is now the leading strand because we're going into the fork. The guy that was the leading strand that we've been working with so long, on the right-hand side, it's the lagging strand because it's going away from the fork. So really one strand is not worse than the other, okay? It just depends on which side of the fork you're on at what point. All right, so what we need to deal with, we've really talked about the leading strand. We got to figure out what's going on with this lagging strand. All right, so let's get into that. This will be the last thing we need to do before you get to actually watch some animations on this. So again, here's our leading strand. We've talked about it a lot. DNA polymerase 3, just tearing this up, okay? So on the bottom, we have to move in the right direction. We have to move towards the right-hand direction, okay? What's being numbered here is not steps, okay? If you want to think about this temporally, like what happened first, start over here. What's being labeled here is the numerical order in which these pieces were put down, okay? So this one was put down first, this one was put down second, this one's put down third, this one is fourth, and it's happening now. The fifth one will be made, okay? Again, temporally, if you want to think about what happened from the beginning, start over here, okay? So we're going out of the fork. Just like the leading strand, a primer must be put down first. So primase does that. DNA polymerase 3 goes out of the fork, okay? But simultaneously, yeah, okay, we cannot add to the 5' end, but things are still opening over here. Like helicase is still tearing through this. There's a lot of DNA to replicate. That leaves a whole patch that we can't go back and get, okay? So what we have to do is then further in, put down another primer and replicate it this way, okay? So it's like as the fork opens, we put down a new primer and here we go, okay? So that's why these are in order. This one happened first. As the fork opened more, we put down this primer and then made this next one with DNA polymerase 3. Okay, if you're wondering why DNA polymerase 1 is here, don't worry about him yet. He's not doing what I'm talking about right now. Okay. Then when we get to the fork opening over here, we put down another primer and synthesize this. When the fork opens to here, we put down a primer and there it goes. Okay, put down another primer. There it goes. We'll put down yet another one, a sixth one. There it goes. Because of this directionality, we have to wait until the fork is open, put a new one, and then keep going. Okay, so that's why this is called the lagging strand, right? So this fork, again, is constantly opening. It's going to be opening until it reaches another bubble, and we have to reinitiate the synthesis, okay? So that's fine, all right? So while the leading strand is undergoing continuous replication, the lagging strand is kind of replicating in shorter pieces. These shorter pieces are called Okazaki fragments, okay? And you may feel like you can't remember this name. You can. This is a Japanese name named after the people that discovered these fragments. Okazaki fragments are the term for these shorter pieces of DNA, okay? They're not going to be short forever, but right now they are. Okay. So again, review that as many times as you need to. It does take a little bit to kind of get the process visualized easily in your brain if it's been a while since you've seen this. Okay, so now we have to kind of finish up, all right, because you notice ribonucleic acid is still in here. These primers were RNA. Okay, we cannot leave them here, even though they look fine, because RNA is very delicate. Okay, it's meant to be a transient molecule. We're going to talk more about RNA and its stability when we get to chapter 18. Okay, we may talk a little bit about it in 17, but it is not meant to hang around. Okay, so maybe it might hang around for an hour. Okay, you want your DNA to last longer than that. Different RNAs hang around different amounts of time, but either way, it's not a super stable molecule. DNA is really much more stable. So what we have to do is replace those primers, okay? Why did we put them in there in the first place if we're just going to replace them? I don't know, man. Biochemistry, because that is how polymerases work. DNA polymerase 3 can't just start wherever it wants, but primase can, so that's what we got to do. Okay, we just work with what we have. We have to replace it. These are replaced by DNA polymerase 1, okay? So this little thyroid-looking guy here, DNA polymerase 1, what it's doing right here is removing the RNA and replacing it with DNA, okay, by adding on to the previous Okazaki fragment, all right? It's still subject to the same rules, so it removes this and adds this on going in the right-hand direction, okay? When it is done, all right, again, no matter what, doesn't matter, in a box, with a box, it doesn't matter. DNA polymerase cannot add anything to the 5' end. Even when it removes this last guy, there's still a gap because it cannot make that bond. It can't create that bond here. So DNA ligase is going to be something that acts like glue to finish up. Once we've replaced that primer with DNA, we seal it together. This continuously happens. It's happening all along. It's happening up here. Anywhere this is, when it meets the next replication bubble, we'll use a ligase. This is going to just seal up all these little like gaps in the actual backbone. So this is going to be creating that last bond. Okay, so think of DNA ligase as a little glue. We'll see that again in the next section. So again, this has been just kind of an overview of what happens with each of these enzymes and components. Talked about the importance of being antiparallel, the leading strand, the lagging strand, and the general mechanics of replication. We talked about it only happens when you're going to divide, okay? So again, I'll have posted for you a second short video, which should be a little bit more animated, should hopefully give you a clear picture of what's happening here. Okay, and then the final video that I will create for you will talk about some proofreading and repair and why it's a problem to be a linear chromosome.